As many of you, I think, already know, my knowledge of English is very, very poor. So all I have to say, you will find written in the slides. What? Oh, yes. Oh, this is done. Ah, change. What? The change is. Yeah. Oh. Yes, yes. In multicellular organisms, fungi, animals, and plants, the morphology is determined by a precise assembly plan, which is practically every detail is the result of a natural selection. This close link between morphology and functionality justify the prevalent use of morphological aspects in taxonomy. In the case of Maxomycetes, on the other hand, the morphology of the sporocarps is not determined by the precise assembly plan, but rather by the outcome of a series of coordinated processes and by the influence of environmental conditions. These processes are certainly the result of natural selection, but not all of their outcomes, often even the most obvious, must necessarily be. This has sometimes led a taxonomy based solely on morphological aspects without an identification of the processes that they have determined them on the wrong path. Oh, no. What? Not. I need a. Oh, with okay. Okay, now. The extensive use of photographic stacking techniques allow to obtain high-resolution optical images of mesocarp. It's true that these techniques, unlike electron microscopy, don't add anything to what direct optical observation can theoretically provide. However, the possibility of extended availability of high-resolution images to many samples can suggest hypotheses on the modalities in which the sporocarp formation processes operate that are not always clearly expressed in literature. Understanding how these processes take place and how they may be influenced by environmental factors is not only of theoretical importance, but essential to distinguish between truly different species and outlier forms. The role played by the presence of lime in the Physarales is one of the most critical areas. We will start with the distribution of lime on the peridium of Physarales. The way in which lime is distributed on the peridium characterizes the different species. In the case of Physarum brunellum, distribution is uniform and recalls that of the genus Diderma. Examining the genus Diderma, Schonacek-Keller stated that the lime reached the surface together with the slime shed, identified as a mucopolysaccharide, through the invagination of the plasma lemma. This lime then acts as a matrix and determines the ultrastructural and morphological characteristics of the deposit. The contribution of the slime shed in the case of Physarum brunellum is evidenced by the lacquered appearance of the peridium and by the detachment of a large fragment of the inner membrane. Yes, you can see. The same characteristics can be observed in the other species, such as Physarum alpestre. An opposite situation occurs in species in which the distribution of lime on the peridium maintains a close relationship with the structure of the capillicium, particularly evident when this is badamioid, this is Badamio utricularis. Indeed, Becht and Horner observed that the lime can reach the peridium also through the outcrop of the vacuoles corresponding to the knots of the capillation. I found the most interesting exemplars by observing the maturation of mixocarps of Physarum decipiens in moist chamber. The appearance of the peridial reliefs is extremely early and reproduce the morphology of the capillation. The characteristics are maintained in the immediate following phase, and it is clear that it is not simply a question of expelled matter, but of the emergence of the outermost layer of capillation. In addition to the lime knots, one can also see the connecting tubular filaments between them. At full maturity, however, the correspondence between the morphology of the external reliefs and that of the capillation is often lost. The reliefs are very prominent and with rounded edges. In order for the reliefs of the peridium to maintain the appearance of the knots of the capillation, the membrane delimiting the emergence vacuoles must maintain its integrity until the consolidation of the contents. What can happen depends on the environmental condition and the consistency and the protective layer offered by the slime sheet, which is probably thinner on the top of the sporotheca. When the membrane dissolves, leaves an opening which the surface tension tends to make circular, while the lime are expelled outwards. The time at which this occurs determines the partial or total loss of isomorphism between the peridium reliefs and the capillation knots. Indeed, in the sample of Physarum fusellum, we find the reliefs which retain the appearance of the badabioid capillation only at the base of the sporotheca. You can see there. In the case of Physarum spectabile, it's the size and density of the reliefs and knots, that demonstrate a perfect match. With such a density of knots, a rapid reduction of the volume of the sporotheca by drying can determine a multi-level outcrop of the capillation. The reliefs are superimposed. Sometimes the situation can be different, and the peridium, the structure of the and on the peridium, the structure of the capillation is not reproduced, even if it is badamioid. Let's see the case of Badamia melanospora. Studying Badamia gracilis, now melanospora, well, then observed that the lime accumulated in internal vacuoles reached the peridium through tubular invagination, constituent parts of the capillation. Indeed, everything may depend on the preferential direction of the development of the capillation and of the distribution of the knots. If you see the upper side, if you consider the upper part of the peridium, we can note that the small reliefs actually correspond to the insertion points of the capillation. With larger deposit, the situation becomes this: no correspondence between peridial reliefs and badamioid capillation. An extreme case is represented by Physarum stellatum, which offers another obvious example of this situation. No relation between the very little knots of the capillation and the relief of the peridium. Emission through the capillation ducts, outcrop of knots, and direct crossing of the peridial plasmalemma are the independent phenomena that can occur with a highly variable relative incidence, even with the same species. Physarum vernum can give significant examples. In this case, the emergence of knots was the predominant phenomenon. You see a correspondence. In this other, the emergence of the knots was followed by an important contribution of the direct crossing, which produced a uniform coverage of the peridium. The genus Fuligo deserves a separate discussion, being distinguished by the cortex covering the peridium. A thick layer of spongy-looking lime that cover often large italia. If one observes the cortex of a Fuligo matured under typical condition, one notices an aspect reminiscent of that of a leavened dough for bread or cakes. The result of a very viscous fluid inside which gas is released. It may assume, it is Fuligoseptica. It may assume that at first a significant amount of degraded protoplasm is expelled from the forming peridium, a layer in which lime is then released. The gas could be a consequence of chemical phenomena occurring inside the ethalium or even the layer itself. Nanenga speaks of the cortex as a crust of sterile tubes, which would imply a structural origin, which, however, does not seem to correspond to what we can see. Anyway, the phenomenon affects the superior cortex and the hypothalamus in the same way. The superior cortex and the hypothalamus. This should imply that the spongy appearance is determined by phenomena which essentially occur within the layers of the degenerated protoplasm. The phenomena occurring in the cortex of the genus Fuligo should not be very different from those occurring in the hypothalamus of Physarales, if you look at the base. The fruiting bodies of Fuligo are often found in very exposed position, where the rapid drying could compromise regular development. The presence of the cortex and the largest side of the etalia can be effective defense against too fast drying. Actually, the same consideration can be applied to the genus Mucilago, which belongs to the Dimiaceae. The protection from too rapid drying. An evolutionary convergence that links Physarum Fuligo to the Dimium Mucilago. On the other hand, it was already known by Lister that in natural or artificial conditions of high humidity, the cortex does not form. This is a Fuligoseptica without cortex. And the fruiting body tends to divide into smaller etalia, which can be reduced to simple plasmodiocarp, like a Physarum. All intermediate situations can be observed. An intelligent reaction to environmental circumstances or unintended consequence on them, drying too slowly also poses a risk. We know the stalk in the Physarales. The stalk in the Physarales, when presented in the same proportion, often has a very similar appearance to what will also be found in other orders, and is usually defined by adjectives such as striate, forward, folded, plicated. In recent high-level work, such as that of Clark and Hoskin on the precipitated species of Didymium, which devotes a substantial chapter of the structure of the stalk, the external aspect is described as longitudinally folded to produce striation. Even not formally explained, it is clear that these characteristics are attributed to the rapid drying of the outer surface of the stalk, without reference to structural elements. If, on the other hand, we observe high-resolution images of calcareous stalks, it is difficult not to think to the presence of structural elements, if you see. What might have looked like grooves and creases instead look like intertwined cords with practically constant section. And it's precisely the fact that these chords appear intertwined and superimposed that makes inadequate an explanation based solely on the effect of rapid drying. Perhaps also in the formation of the stalk, there are phenomena not too different from those involving the peridium and the capillation. It's reasonable to think that the mechanism underlying the mobility of the cytoplasm in the formation of the spore cap are the same ones which allow the mobility of the phaneroplasmodium in the vegetative phase. A very clear presentation of the structure of the main branches of the phaneroplasmodium was given by Wolfgang Botterman. In the center, we find the cytoplasm which pulses axially, while the periphery is massively affected by invagination with the digestive and excretive function. If we imagine this structure verticalized, everything may seem clear. The pulsation, here unidirectional, push the cytoplasm upwards to form the sporotheca, leaving the stalk like an increasingly narrow tube. If the invagination are free of lime, may be easily reabsorbed, whereas if they contain some, they could maintain a trace of their presence. There is no reason to think that the remnants of the imagination cannot overlap or intervene, but their original submicroscopic diameter will be much smaller of that of the cores we have identified. To reach the size of the cores, maybe we could think that the imagination have become chains of elongate vacuoles. The phenomenon highlighted does not only occur in Physarum, but also in the demium of the squamulosum group, substantially in the same way. In their classification of the stipitate species of the demium, Clark and Hosking, they placed the demium squamulosum in the empty tube externally calcareous section. This would compatible with the hypothesis that the residues of the invagination are the almost exclusive recipients of the lime in this type. Apparently, what is observed in Physarum and Physaracea and in Dimium and Didimiacea does not seem to be true for the Derma and other Didimiacea. If you look at the stalk. In this case, indeed, an explanation based on wrinkling resulting from rapid drying would seem sufficient. However, cores related to invagination should have a smaller section and therefore be much less evident on larger diameter stipes, as are often found in the Derma, Derma montanum. On the other hand, it is clear that in the case of the Derma, the most external part of the stalk is not the privileged destination of lime. The Derma hemispherical can often give an obvious and very particular example, a cartilaginous tube filled with washed material. The lime inside the base of the stalk appear as large crystalline grain. A recrystallization probably due to the presence of water expelled from the cytoplasm during the formation of the sporotheca. Anyway, it is not an occasional phenomenon because it occurs very often, both in the field and in most chamber. The base. It is regularly found even in much more exotic species, and one can observe it by the transparency of the outside free of lime of the stalk. This is the Derma punense. The size of the granule that we have seen at the base of the stalk are almost equivalent of those that we can observe in the columella of sessile species, is the Dermatistaceum. It's probably no coincidence that the size is also quite similar to that of the cords we saw earlier on the stalk, and of almost the knots of the capillation of many Physarum, perhaps the medium size of vacuoles collecting lime. Bertolone related the presence of calcium ion to the mobility needs of Plasmodium and found that the production of lime always occur after this need have been exhausted. In stipitated species, they can therefore only occur when the sporotheca has differentiated from the stock. In some cases, it happened that the base of the stock is already consolidated before lime production have been started. So we have no lime at the base. The phenomenon is particularly common in the Dimium squamulosum group. But it also can happen in Physarum with calcareous stock, like Physarum melium, base without lime. It seemed that simple delay in the production times of calcium carbonate could make the difference between species with calcareous or limeless stock. But what can happen if instead of a delay, we should have an advance? We consider the Dimium minus a species that should develop a calcareous hypocolumella only after the complete formation of the stalk. Is it the hypocolumella? Sometimes the limes is also released in the upper part of the stalk, which is strongly enlarged. Maybe an advance in the timing of the lime production. When this phenomenon occurs, they extend to all sporocarp of a fruiting, and it is therefore probable that they have a genetic basis. Most of the studies on the processes that led to the formation of most sporocarps date back to the 1950 and 1970, and were also conducted with very sophisticated techniques. Currently, this is not a leading research area, even if it is clear that genetic affinities between species must be reflected in affinities between these processes. Also, this presentation, various hypotheses have been proposed on the mechanism of mixocarp formation. It must be clear that the intention has always been to highlight the problems, and certainly not to identify solution which will be sought by different means. Thank you. Okay.
So
Please no question.
Okay, if nobody wants, then I simply ask, what would be the function of the lime? One is obvious, it's stabilization of the stock.
We know with the lime is with calcium is present in a large amount also in another order, like metatrichia.
Yes.
But it's not transformed itself in lime. So
But could it be that at least in Physarales, the lime is also regulating the speed of outdrying, how fast the fruiting body dries out?
okay, thank you.
I would like only to comment, and from your presentation, it is obvious that when we look for some new characters, morphological characters,
Sorry.
they are not only stable characters, but maybe we should look also on development of the characters that can differentiate the species or even genera.
Yes,
so this
yes.
is a very, very interesting perspective.
Yes, I think,
So,
I think
the development that can
the processes are
Yeah.
most important than their outcome.
Yeah.
The outcome but depend on the environmental condition.
Yeah.
The important is the processes that produce the
Yeah.
sporocarp. Yes, involved in the production of the sporocarp.
Yeah.
Yes.
Yeah.
You obviously studied a lot of species. Did you study other species of Fuligo, like Levidarma, the ones with the smooth cortex? Levidarma levis, Luteonitens? Fuligo?
No.
No, you just Fuligo septica.
Yes. Yes.
Okay.
You know, a few
Because
species.
it's quite different
Yes.
from the others.